All right. Nice to be here. Drugs and how do we get them faster? We need to make medicine discovery computational if it's going to speed up. I came here I came here to SciFood 2010 to talk about open source drug discovery, so how we do Wikipedia for drugs, essentially. And we've been doing that for the past about 20 years with these rules and searching for good components and ways to do drug discovery without any secrecy, including lab books and using GitHub and social media and things like that. And in that time, we've shown that we can speed things up by being open, because then you get really good contributions from students and the public and executives in the pharma industry, and everybody wants to help if you make it open. So that's been going really well. The problem with drug discovery is it takes so frickin' long. So to find the beginning of a drug discovery project, in other words, a small molecule that binds your protein of interest, is really hard. It takes ages. If we're going to speed up finding important medicines, we need to put that bit into a computer. At the moment, every undergraduate will tell you that if you put a protein into a computer, of course you can predict a molecule that binds it. Well, they're wrong, and everyone who says that is wrong. You can't do that yet. So you can't take a protein, put it in a computer, and find a small molecule binds it with a computer. Sorry if that's news. We need to make this computational in a way that we've seen already, right? AlphaFold is a fantastic tool, but was built very cleverly on decades of high-quality experimental data, put in the public domain by hard-working scientists over many years, right? Openly available, high-quality, curated, machine-readable data. And what do you know? AlphaFold comes along and transforms it. We need this, but for small molecule protein interactions. In other words, the beginning of every drug discovery project. How do we do that? Well, this really nice paper came out about 4 years ago, led by a guy called Patrick Riley. He used to work at Google when they did this work. It's a great paper and not cited enough. Right, so this is a way of doing a screen against, of billions of molecules against a protein using something called DNA-encoded library technology. They didn't invent DEL, DNA-encoded libraries, but they use it in a clever way. So DNA-encoded library is, you can have a small sample that contains billions of molecules, and every small molecule is attached to a string of DNA. The DNA is like a barcode that tells you what the molecule is, right? So what you do is you incubate billions of molecules at the same time with a protein, and you filter out the binders, and you sequence the DNA. And that tells you, of the billions, these few million bound, and these other gazillion did not bind, right? Amazing, right? So you just got to do a lot of sequencing, but it's amazing. The really fun bit, which is what this paper is all about, is that you can use those data, the binders and the non-binders, so the positives and the negatives, to build a machine learning model that allows you to predict molecules that will bind the protein. Crucially, the molecules that you're predicting are ones that you can buy now from companies like Enamine in Ukraine, who sell molecules very quickly, right? So I can rock up to Enamine, they've got a library of 40 billion molecules, I can buy one of them and it arrives in 2 weeks' time. So in this case, you can have this massive library, develop a machine learning model, and you can use that to predict a molecule that you can buy. So you can do the whole thing in like a month. It worked in this paper, and everyone was very skeptical about whether this could possibly work. This DEL ML thing, DNA-encoded libraries plus machine learning. And so with colleagues at the Structural Genomics Consortium, we've been messing around with this for a little bit in the intervening years, looking at proteins, this beautiful thing on the top right called the WDR protein. It looks like a plate. And they modulate protein-protein interactions. And it turns out using this approach, so screening this big library and predicting molecules you can buy, has worked about half the time to give like a low micromolar binder, which is enough to start a drug discovery campaign. So that's very exciting. The machine learning is tricky because the molecules attached to the DNA don't look anything like the molecules you can buy. So you're developing a model on a bunch of molecules that don't resemble the things you can buy. That sounds like a nightmare to me. But the machine learning people we speak to say, oh, that makes it really interesting. So that's good. All right, so we've done the pilot and now the big project. So the thing which is going to lead to the AlphaFold equivalent for predicting small molecules by proteins will happen in the next 5 years. And so I'm part of an initiative with the SGC to do this on scale. So the idea is in the next 5 years we make 2,000 proteins, which we can easily do. You screen them in the DNA encoder library, you do the machine learning, you predict molecules, you test them in the lab, and that cycle then means that the machine learning models get really good. So we are looking, essentially, we're going to generate the vast amounts of experimental data. What we're looking for is machine learning people to help us make this a reality. So I'd love to talk to you about that.